Good afternoon. I think we might make a start. It's my pleasure to introduce Daneika Chandrananda, who is, I'm the primary supervisor of Daneika. It's been an absolute pleasure to have Daneika as a PhD student. She came to me on a personal recommendation from a friend from Auckland. So she's actually a New Zealander, and I have a couple of New Zealanders in my lab, so that's quite good. Daneika really, to me, epitomises what a PhD is about, because she's really shown great tenacity to get where she is. And today is not really a case in point. Again, Daneika's had the flu, so she's not feeling all that flash, but I'm sure she will battle through this. Daneika's work has been about the analysis of cell-free DNA data with applications to detecting fetal aneuploidy. And like many PhD students, she's definitely come to the point now where she's the master of the subject, and discussions with her are quite difficult for Natalie, who's also sitting there, who's her co-supervisor. It's clear now that Daneika is the expert and we are her disciples. So that's good news, I think, for any PhD student. Yes, so without further ado, I'll let Daneika tell us about her PhD. Thanks.
Hello, everyone. Thanks, Melanie, for that really kind introduction. So I thought I'd start by giving you an introduction into cell-free DNA, because that is what my PhD is built around. So this cell-free DNA exists as these short, double-stranded molecules, and you can find it in various body sources, such as human plasma, urine, cerebrospinal fluid, and synovial fluid. And these fragments don't exactly exist as naked DNA, but they are protected by being bound to histones and being in vesicles, as well as being inside lipoprotein complexes. But there really isn't a lot of understanding around what protects this DNA. Now, there's been several studies that give evidence for these fragments coming from apoptosis, necrosis, and even that they are actively secreted from living cells. Now, all of us have cell-free DNA in the different sources of our body, but in some cases, you get a mixture. So, for example, in pregnant women, a little bit of fetal DNA that's chopped up starts circulating in the mother's plasma. So you get a maternal-fetal mixture. And in the same vein, if you've got an organ transplant, then you got a little bit of the donor's DNA mixing in with yours. And in the cancer setting, you get the tumor-normal mixture. Our lab and our collaborators are interested in sort of this mixture scenario, although my PhD revolves around this maternal-fetal scene. So a little bit about that. The percentage of fetal DNA in a pregnant woman's plasma varies according to how far along she is in the pregnancy. So in early pregnancy, you get very little, and as the pregnancy goes on, it increases. So there's a range about from 3 to 20%, although sometimes there are some samples where you see up to 50% of the mother's cell-free DNA is actually of a fetal origin. And this percentage has an inverse relationship with maternal weight, where they've seen women with high BMI showing a low fetal fraction. So fetal DNA can be accessed by a simple blood sample. So it's a fairly non-invasive way of getting access to fetal DNA. And it's also been found that once the woman gives birth, within 2 days, there is no trace of the fetal DNA in the mother's blood. So how does this extracellular DNA tie in with prenatal testing? So the main reason for prenatal diagnosis is the need to detect Down syndrome. Now, Down syndrome is also known as trisomy 21, and it occurs because of an extra copy of chromosome 21. So instead of 2, you have 3 in the fetus. And this leads to severe developmental and intellectual disabilities. And it's actually the most common chromosomal aberration occurring in about 1 in 700 newborns. And the gold standard and definitive diagnosis of this condition is to collect fetal cells directly. You can do this by techniques such as amniocentesis or chorionic villus sampling, but these techniques are invasive and can cause miscarriage. And there has been a lot of interest to search for a non-invasive way of getting a definitive diagnosis. So that's where this cell-free DNA mixture comes in. So fetal DNA was actually first detected in maternal plasma way back in 1997. And throughout the years, there's been a concentrated research effort into finding how to use this mixture in prenatal testing, but there has been several setbacks. So first the researchers started focusing on, instead of the fragments, they tried to see whether we can get intact fetal cells. But then they found out that it's quite rare and that fetal cells persist in the mother's body even a decade after giving birth. And so you don't want to confound 2 different pregnancies. And then they started looking into separating or enriching fetal DNA from the maternal DNA. And they found that they couldn't get one technique to be robust enough to translate into the clinic. So it wasn't until around 2007, 2008, when next-generation sequencing really started coming into play, that they looked into NGS as the technique to interrogate the fetal DNA and then translate it to the clinic. So the very first test that used whole genome sequencing and this cell-free DNA mixture to test for trisomy started coming up from 2011. Now there are about 5 or 6 tests out there, mainly developed in the US and about 2 in China. So that the first section of my PhD project has been around this. So it's collaboration with the Victorian Clinical Genetics Service, which sits at MCRI. They wanted to have a local non-invasive trisomy test. And the reason for this was that having bioinformaticians who fully understand the pipeline meant that we could put in any improvements we needed, and we had the flexibility to change the test however we wanted. And of course, if everything went well, you could offer the test to the women of Australia at a lower cost. Because right now, the NIPT tests cost something from $800 up to $2,000 US. And of course, it gives us the flexibility to extend this test. So instead of just looking at whole copy number changes, we can look at subchromosomal insertions and deletions. And of course, doing work like this from ground up means we have a better understanding of cell-free DNA, and this knowledge can be used for other projects that our collaborators are interested in. So I'm going to take you through a standard pipeline that uses whole genome sequencing of cell-free DNA in maternal plasma to detect trisomy. So once you get a blood sample from the pregnant woman, in order for the fragments to be sequenced, it needs to be prepared into a library. So maybe a lot of you have actually got experience in a next-generation sequencing project that uses cellular or genomic DNA. So the main reason, the main stage at which the way you process cell-free DNA and cellular DNA differs at this library preparation. So in the library prep, you have to first extract the DNA. In cellular DNA projects, you get this from the nucleus of white blood cells, for example. And it's a fairly standard procedure, and you get sort of intact DNA. For cell-free DNA, you need to first separate out the plasma from the whole blood cells, and then you need to separate out the fragments from the plasma or from the other things in the plasma. And there are several dedicated kits that needs to do this because it's quite an intricate process. In cellular DNA, you need to fragment up that intact chromatin in order to be sequenced. So they use methods such as sonication or nebulization in order to randomly cut up the DNA. Cell-free DNA is randomly chopped up. It's naturally chopped up. Therefore, we don't want to cleave it further. Therefore, this step is skipped, and therefore you get sort of the biologically cleaved fragments. Next, in cellular DNA projects, once you randomly cleaved your DNA, they select a certain size range, and that depends on your project. For cell-free DNA, the majority of the fragments are quite small, so less than 200 base pairs. So once again, this step is also skipped because we want that full size range. The polymerase chain reaction is something that is routinely done in order to amplify your DNA, in order to go into a sequencer. And for cellular DNA, you use a standard number of PCR cycles. Cell-free DNA, you need to actually have a higher number because you start with a low starting amount of DNA. And once you've got your libraries prepared, you can just put it into a sequencer. So what you get out of a sequencing machine are these reads. So a read is just the first number, a certain number of bases of your DNA fragments. So it could be 50 bases or 35 bases and so on. And then it's up to the bioinformaticians to map it back to the genome, etc. Now, there is no difference in the way we analyze cell-free DNA compared to cellular DNA in this part of the pipeline. So you do standard alignment and standard read processing. So in the commercial pipelines, what they do is they use an aligning software to map each read to the human genome. So they basically find out where each read comes from. And sometimes when a read comes from a repeated region, you cannot say exactly where it maps to because it can map to multiple places. And therefore, they discard these multi-mapping reads. There's also filtration of reads that are PCR duplicates, which is a usually standard step. And then they filter on mapping quality criteria or some sort of removing reads that have a certain amount of mismatches. So once you find out where all your reads come from, you can calculate a count for each of the chromosomes. So in this little cartoon, you see the counts binned according to the chromosome. So in real data, you have millions of reads. So what we are looking for is the trisomy signal. But right now, all our counts are still in this maternal-fetal mixture. You can't tell whether one read is from a mother's fragment or from a fetal fragment. But if you get a blood sample from a trisomy 21 pregnancy, you would expect a slight increase in the amount of reads that map to chromosome 21. And if you compare it to a normal sample, you can detect this increase. So the way they do it in the commercial tests is that they find a proportion for the chromosome of interest. So for example, because we're interested in chromosome 21, you can get the read count for chromosome 21. That's basically what maps to 21, divided by the total read count for the sample. So that is your proportion for your test sample. And then you can compare this proportion to the average of normal samples via a simple Z-score test. So basically what you do is you get that proportion, subtract the average of a normal set. So these are references which come from pregnancies with normal, non-trisomy fetuses, and then you divide it by the standard deviation of the normal set. It gives you a Z-score for each of your test samples. A Z-score is basically, you can think of it as a distance away from the average of your normals. So basically, that is what a standard pipeline looks like for non-invasive prenatal testing. But this works in theory if the only noise in your counts or the increase in your counts comes from having an extra copy of chromosome 21. But in truth, next generation data, it's pretty noisy. So you can't just simply count the sequences and do this test. The noise will cancel out your trisomy signal. So there's several different sources of bias that affects the counts at each genomic location. One of the most well-known biases is GC content. So if you've got really high GC content or really low GC counts in your genome, in the regions, you tend to get a relatively low number of counts. The second source is mappability bias. Now, this goes back to the point I made earlier. In repeat regions, you get low number of counts. So in repeat regions, you get low mappability and you get low counts. The third source of bias hasn't been investigated very well, and it's known as fragmentation bias. This occurs if your fragments are cleaved at specific sequence motifs. So if the fragments tend to be cleaved at this sort of AAA motif, you won't get the same amount of counts at a CCC motif. So that is another source of bias that affects your counts. Now, the commercial tests actually account for some of these biases. To correct for the bias due to GC, they do an empirical distributional adjustment, and for the bioinformaticians, it's simply this lowest correction. And to account for the bias due to repeats, what they do is a hard filter where they filter out all genomic regions that are designated as repeats, and along with it, any count that goes into those regions. And the third source of bias was not documented in cell-free DNA until we started working on it and started seeing this bias in our data. So none of the pipelines actually account for this bias. So we decided that our local pipeline could do better, and we wanted to see whether we could have alternative steps at these, to correct these different biases. To correct the GC, we decided to adopt this method by Terry and his student. It was first introduced for cancer whole genome sequencing data, and it's based on a model. So what we did was to incorporate properties of cell-free DNA fragments, such as length, and to make it more tailored towards cell-free DNA. To account for mappability bias, we did not use a hard filter. We quantified the mappability in each genomic region and corrected the counts according to that mappability. But this wasn't enough, because in next-generation sequencing data, you sometimes get stacks of reads that occur because of the nature of that sequencing. And these stacks of reads occur at these blacklisted regions of the genome. So the ENCODE project has got some nice annotation that gives about 226 small regions that have these huge numbers of reads. So once we do the mappability correction, we actually remove these special regions of the genome. We also assess the fragmentation bias using the model specified, and we implemented a correction for it. And what we found out was once we accounted for the GC and mappability bias, this fragmentation bias, correction for the fragmentation bias did not actually do any further improvement. So what we did next was to run a small pilot study to compare our local pipeline with what is standard in the literature. So the datasets we've used come from 29 pregnancies. 9 of them had confirmed Down syndrome fetuses, and 20 were normal samples. So all of these, the cell-free DNA, they went through whole genome sequencing on a HiSeq 2000 machine, which spit out single-end 50 base pair reads. So we started with an average about 26 million reads per sample. So this comes to quite a low coverage of 0.42-fold average coverage per the genome. And then what we did was we sampled the starting number of reads to simulate lower and lower coverage. We went from 26 million on average down to less than 1 million. We did this to see whether the pipelines could still detect the trisomies with very extremely low coverage. This was important because these methods were going to be clinically translated. And if you have low coverage, it means faster turnaround time as well as a lower cost. So these are the coverage statistics. As I said before, the original number of reads was around 26 million. And then we sampled 75%, 50%, etc., and got it down to less than 1 million reads. So that was an average coverage of 0.14 fold. So when you first run the data through the standard bias correction pipeline in the commercial tests, this is what you get. So on the y-axis, you've got the Z-score for chromosome 21 for the 29 samples, and on the x-axis, you've got the coverage. So this is for the starting coverage. The bar here stands for Z-score of 3, and this is the diagnostic threshold used for trisomy detection. So at the starting coverage, the standard bias correction pipeline detects all of the trisomies, which are the 9 triangles, and all the normal controls, the 20 normal controls, are below the threshold. Then as you decrease the coverage gradually, you can see for the second level of sampling, you still detect the trisomies at 100% sensitivity, but when you decrease the coverage further, you start getting your first false negative, and then it's sort of downhill from there onwards. When you look at the local bias correction pipeline, for the first starting coverage, we also detect all of the trisomies, but there is a higher discriminatory distance between the trisomies and the normals, which means we won't get sort of samples at the boundary which can be misclassified. Then as you decrease the whole genome coverage, you can see that we still detect the 9 trisomies right up to this coverage. So this is extremely low coverage. It's about running 10 samples in a new MySeq machine. And you still get a pretty good discriminatory distance between the normals and the trisomies. So this meant that by using improved bias corrections to control for the noise in the next generation data, we can still detect the trisomy signal by even using this extremely low kind of counts of data. Because we ran this pilot study with a relatively small number of samples, BCGS wanted to test this on a larger cohort. So what they were already recruiting samples from pregnant women who wanted to have the NIPT test done. But what they were doing was they were taking one blood sample and sending it off to the US and taking another blood sample and running it through our local pipeline. So from the first 18 samples, we detected all 3 of the trisomy 21s, which also had diagnostic tests done on them. And then we also correctly classified one sample as a normal, non-trisomy, while the commercial test classified is as a trisomy 18. So these tests aren't just chromosome 21 specific, you can actually use it for other chromosomes as well. So this is an ongoing process, and I'm currently working on implementing sort of a semi-automated pipeline over there. And we're really happy that our method was also picked up by an NIPT group over at the UK, who have also collected about more than 1,000 samples, and they want to see how the method works on a really large cohort. Right, so that brings us to the second section of my talk. And this is where we started investigating the features specific to cell-free DNA. The motivation behind this was that we started observing some really interesting signals when we were looking at the low coverage data. And when we went back to the literature, we couldn't find anything about what we were seeing, because this seems to be due to a lack of basic research done in cell-free DNA, because this whole arena had been led by the prenatal diagnosis groups, and they were funded by the industry, and they were always looking for any method to be clinically translated. So even though next-generation sequencing has an extremely high resolution, there was a push for lower and lower coverage, and therefore they sort of lost the opportunity to interrogate the cell-free DNA. So there's a lot of holes in the literature that now seem to be holding back biomarker development for in other areas. So our aims in this section was to find out about as much as we could about cell-free DNA features using high coverage data. So we wanted to see what the lengths were in these fragments, and to see whether they were cut up at specific sequence motifs, and where they came from in the genome, whether they came from specific places in the chromosomes, etc., and basically document how cell-free DNA differed from cellular DNA. At the end of it, we wanted to get an understanding of how cell-free DNA looked like in a normal person. So we couldn't generate extremely high coverage data locally, so we looked for public data sets, and we found one published by Jacob Kitzman, which was first published way back in 2012, although the data wasn't released until last year. Now, what they did was they used this data for something completely different than what we used it for, but we were very lucky to find it because this data kind of fulfilled our wish list. So the data came from 2 pregnant women. They will be named I1M and G1M. So both of these women donated the cell-free DNA from the plasma and then the cellular DNA from the nucleus of lymphocytes. These pregnancies had normal male fetuses, nothing to do with trisomies, and they underwent 101 base pair paired end sequencing. So our low coverage NIPT data was 50 base pair single end sequencing. So this is the first time we actually had data from both ends of the DNA fragments. And also, the data came at a extremely high coverage of 75-fold whole genome coverage, which amounts to about 2 billion reads for cell-free DNA in I1M, 1 billion reads for cellular DNA, and then a little over 1.5 billion for G1M cell-free, and then 850 million for cellular DNA. And once again, I need to remind you that the main difference between how cell-free DNA and cellular DNA is processed occurs at that library prep step, where cellular DNA is randomly fragmented and size-selected, while cell-free DNA is not. So the first feature we looked at were the fragment lengths. So here you've got the fragment lengths on the x-axis and the density, or sort of a relative abundance criteria, on the y-axis. And then you've got the two samples, I1M and G1M. So this is the profile of the autosomal fragment lengths for cellular DNA. So you see you get this one peak in a fairly narrow range between 100 and 250. The thing is, cellular DNA, like I told you before, has been size-selected. So if the original investigators wished for it, they could have just slid this distribution along this scale, but they have chosen this size range. When you plot the cellular mitochondrial fragment length, it follows the same profile as the autosomal length, which means that there is no, they are not subject to two different processes to get to this point. When you plot the fragment lengths for cell-free autosomal data, you can see it's quite different. We actually get 3 peaks. The first one is over here, the second one is there, and the third one is a really tiny peak that you can't really see. And when you plot the cell-free mitochondrial fragment lengths, it's a completely different profile altogether. So I'll just discuss these differences and any biological relevance to them. So this is a plot of the autosomal cell-free fragment lengths for one sample. What's really interesting is that the lengths in the first peak correspond to the length of DNA that is wrapped around a nucleosome. And the second peak corresponds to the length of DNA that's wrapped around 2 nucleosomes, plus that linker that joins them up. So that's mononucleosomal DNA for the first peak, and then dinucleosomal DNA for the second. And for the third peak, it corresponds to roughly 3 nucleosomal lengths. So when you plot that mitochondrial DNA, you get this extremely variable size range, and that nucleosomal subunit signature is lost. Now, this sort of makes sense because mitochondrial DNA, unlike the other chromosomes, are not packaged as nucleosomes. So they exist as these circular structures, and therefore they are more exposed to cleavage. So this gives us an idea that cell-free DNA in the chromosomes appear to be wrapped around nucleosomes and are protected by that histone structure, and that they are primarily cleaved at these exposed linkers. When you zoom in on that first peak, you see that you get this periodicity up to a certain point. Now, this periodicity corresponds to 10 base pairs, and it seems to be due to that helical, the distance between a helical turn of the DNA. So we can think of this as DNA being wrapped around a histone, so that one face of the DNA is protected by the histone, so that if an enzyme needs to cut at this DNA, it needs to it's only able to cut at this distance, which is a multiple of 10 base pairs. Now, this gives us some evidence that first the DNA is cut at the linker, and then it's cut into the nucleosome at this sort of the 10 base pair periodicity. But if we go back, we see that the most protected structure is that mononucleosome, the single nucleosome structure, while there's only a few fragments with these leads, which means once the nucleosome starts unraveling, the DNA is not protected and becomes rapidly cleaved. Those profiles I showed you before were genome-wide profiles. Now we wanted to see whether there was a variation in fragment length in the genome, whether long fragments came from certain specific regions as opposed to short fragments, etc. So first of all, we looked between chromosomes. We wanted to see whether the proportions that belong to that first, second, and third peak were the same between chromosomes. And we used a mixture model set up to do this. And what we found out was that the proportions that belong to that first mononucleosomal peak was the same between chromosomes, and it was the same for the other two components as well. There were no obvious differences. But when we started looking, going along the chromosome, we started noticing some interesting signals. So we narrowed down our analysis to look at the differences of fragment length in the repeat regions. And today I'm going to just introduce what we've seen in fragments that come from microsatellite repeats. So in black, you get that genome-wide profile you saw before. And in dark blue, you get the fragments that map to microsatellites that have this repeating subunit, GAATG. And in light blue, you have fragments that map to microsatellites that are designated as CATTC. So you can immediately see that the fragments that map to these two microsatellite classes have generally shorter fragments than what is expected. Now, there are many different types of microsatellites, but this sort of shifted distribution only occurs at these two types. So it seems to give us some insight that any sort of nucleases or biological processes that cleave this cell-free DNA seem to have a preference for these two types of motifs. But as bioinformaticians, there's nothing we can further do to interrogate that, just sort of make up a hypothesis. All of the plots I've shown you before still comes from the mixture, so we have not separated out the fetal and the maternal DNA. Now, we can actually do this in silico because of the experimental setup of our data. So for both I1M and G1M pregnancies, we've got the cellular DNA sequenced, which is just maternal information. And we've also got a cell-free DNA sequenced, which has that mixture, maternal and fetal. If you use the reads to genotype on known SNPs, we can find out the SNPs which are homozygous in cellular, so where the mother has sort of the AA genotype, and then find SNPs, the same SNPs which have a heterozygous genotype in cell-free DNA, which means the fetus is bringing along the B allele from the father. So at these informative SNPs, we know that the fragments that carry the B allele are actually fetal, and then we can separate them out and then assign the shared allele as the mother. So for I1M, there were 26,000 informative SNPs, and for G1M, there was just around 7,500 informative SNPs. The fetal fractions were different among the two samples. I1M was further along during gestation with 18.5 weeks, so it had a fetal fraction of 13%, while G1M had 7%. And we actually calculate this using a mixture model as well as counting alleles. So when you plot the fragment lengths for the maternal component and the cell-free fetal component separately, you can see that cell-free DNA in the fetus is shorter than its maternal counterpart. This seems to be because either there's a second stage of cleavage happening for fetal DNA, or that fetal DNA is not packaged as securely as maternal DNA. Now, other groups have also noticed this difference, but currently there is no understanding as to exactly how this happens. So I'm going to switch focus right now to a different feature in cell-free DNA. This is where we actually looked at sequence motifs at the cleavage site. So we wanted to see whether cell-free DNA was cut up at specific sequence motifs, or whether it was independent. To do this, we went back to the mapping positions of all of our reads and found the base at the cleavage site. So at the 0th position, we just found which base was there for each of our fragments. And then for the first position into the fragment, second, third, and -1 means the first position outside the cleavage site, -2 is the second position outside the cleavage site, etc. So for every read, we just found the bases at all of these positions. And then for each of these mononucleotides, we calculated the proportion of fragments that supported it at each of these positions. So for example, this point would mean give the proportion of reads that had base A at the 0th position, and this point would be the proportion of reads that had base A at the first position, etc. What we were interested in the sort of the marginal profile of the 4 bases. So I'm going to show this profile for cellular DNA first. So on the x-axis are the positions. The dashed line is at the 0th position, which is the cleavage site. And on the right, you've got the positions going into the fragment, and on the left, you've got the positions going away from the fragment. So for cellular DNA, you can see that we know that it's fragmented randomly at library prep, and there doesn't seem to be any sort of large preference for any base at any position. Although there's this sort of a small preference for the cytosine base at position -1. But I found out that this is a bias by the Covaris instrument that was used to fragment this DNA. So in general, there is no preference for a certain base at a certain position. When we look at the matched cell-free DNA for the same sample, you see a completely different profile for each of the bases. So if you look at it a bit closely, you can see that this is a very position-specific pattern, and it extends about 8 to 9 base pairs on either side of the cleavage site. And cytosine seems to be preferentially wanted at the 0th position, the first position, and the -2 position, while thymine seems to be preferentially observed at the -1 position. And when we looked, this was looking at the 5' side of the fragments, and when we look at the 3' strand, it just gives us the exact reverse complement. So this is just for one sample. I wanted to show you how reproducible this pattern really is. So once again, that's the profile for the high coverage data that we've downloaded, and these are the profiles for the 29 low coverage sample. So the pattern actually comes up with that lower than 0.5-fold coverage data as well. So the solid line here is the average of the 29 samples, and the dashed lines above and below that are the minimum and maximum proportions at each position. And so you can see that the minimum and the maximum actually follow the average really well, and there's really very little variation between the samples. And we've looked at these profiles using different library preps, and these are across different sequencing centers and machines, and it's actually, when you look at the difference between the two types of data, there's very little difference in the profiles. So once again, these profiles are from the autosomal fragments. So I also wanted to see how it compared to in mitochondrial DNA. When you plot the mitochondrial DNA profile for cellular data, what you see is that you get the same profile, but it's at a shifted proportion for each base. And this is because mitochondrial DNA, it's double-stranded, but it has one strand, which is called a heavy strand, which is cytosine-rich, and that tends to change the average proportions of the other bases as well. But overall, you still have that cytosine preference at this position, and there doesn't seem to be any major difference between autosomal and mitochondrial DNA. In contrast, when you look at the cell-free data, you get something different. So mitochondrial DNA is the lighter colored line, and it does not follow the pattern shown for autosomal DNA, just like in the fragment length. Although there is a preference for cytosine at that cleavage site, but other than that, it does not follow that overall pattern for any of the bases. So this is another feature where being associated with the histone changes what we see in the data. And this could also be related to how the two types of DNA are cleaved, or the biological process that gives rise to these cell-free DNA fragments. So we kept on going, analyzing this high coverage data, and we looked at other features as well. We looked at the difference in the representation of different types of repeats between cellular and cell-free data to see whether certain types of regions were protected in the cell-free DNA. For example, we found that ALU repeats were more highly represented in cell-free DNA, and so were microsatellites. We looked at the relationship between fragment length and GC content, the relationship between fragment length and cleavage motif, and we were also very interested in looking at the enrichment of reads along the genome. We wanted to see whether we got stacks of reads that corresponded to chromatin state. For example, open chromatin and then sort of closed chromatin, or nucleosome-bound positions. And we also want to see whether there's any difference in the features between hyper and hypomethylated regions. So that by using this high coverage data, we now have a better understanding of the natural landscape of cell-free DNA. And we also managed to get together some techniques that allow us to summarize these features genome-wide, which allows us to compare between samples and compare between different states. And the lab is interested in seeing how these features differ between cell-free DNA that comes from urine, cerebrospinal fluid, and plasma in sort of a three-way comparison. And our collaborators are especially interested in the donor-recipient mixture, and they want to see whether the features that I've discussed before are different when someone who's had an organ transplant is rejecting the transplant as opposed to the transplant being stable in the body. And we've also identified how we can incorporate the cell-free DNA features into our bias corrections for the trisomy testing work. And we've got the analysis machinery set up so that we could do any form of comparison to distinguish any diseased and non-diseased state. That brings me to the end of the talk and to the acknowledgements. So I want to thank Howard, Damien, and Daneika. These are our collaborators from VCGS and MCRI. It's been a really successful collaboration, and they are really great people. We've had a lot of interesting discussions from them, especially about the wet lab side of cell-free DNA work. I want to thank Yuval for helping out with the GC bias correction work. And I want to thank Keith, who is not here, for helping out with all the IT-related issues that come from analyzing whole genome data. And these are my funding sources. This is the most important slide. I really want to thank Melanie for being a fantastic supervisor. I think I've learned so much from doing this project under you from day one, from the discussions we've had and just watching how you do science research. So I want to thank you for everything you've done during this very long journey. And also, Natalie, I've enjoyed working with you. I think you're one of the most optimistic people I know. You give the best pep talks if I'm feeling down due to some personal issue, if I'm hitting some sort of roadblock in the research. And I don't think I'll ever forget your mantra of dig, 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 and keep digging into the data. I also want to thank Terry. You were a great help during the birthing pains stage of my PhD, and helping me out with the GC correction stuff and for all the time you've given for the discussions we've had. Thank you very much. And I also want to thank WEHI Bioinformatics. I think this is a great division to do a PhD in. People are friendly, they're very good at what they do, and they're very helpful. And I'm really going to miss you once I finish this PhD and go off to wherever. So thank you, everyone, for listening to this talk, and I'll take any questions you might have.
I have two related questions.
Right.
So in your trisomy studies, if you had a larger enough sample, what would predict you should be able to figure out for each of the numbers of reads, you could predict false positives and false negatives. So you could guarantee from your assay, if you had 1 million reads, the percentage of false positives would be this, the percentage of false negatives would be that. And that would help you have confidence in the assay depending on how the test read out, right, in terms of number of reads, because to begin with, you don't really know.
Right.
But having said that, if that were the case, I want to know the variation between the fetal to maternal cell-free DNA. Is that just individual variation, or is there longitudinal variation? So if you took a blood sample from that same person a week later, might you find more, fetal DNA?
Right. Okay. So to answer the first question, that the trisomy signal is actually dependent on the fetal fraction, and we don't know that information for each sample we get. So so we would have confidence for a sample with a really high fetal fraction, but not for one with a low fetal fraction. And the commercial tests actually don't want to detect, don't want to run the sample for anything that has less than 5% fetal fraction. And to answer the second question, the studies they've done, it's been in larger gestational windows than a weekly basis. So for the, they've compared between the first trimester, second and third, and then you sort of get that linear relationship of increasing fetal DNA, but not in those sort of very close time points.
But did they look at the changes? So maybe there were changes in the, like the motifs and the fragment lengths. That wasn't looked at.
No, it wasn't looked at. No.
So a couple of questions. One was you mentioned PCR duplicates. What do you mean by that? Are you talking about prime diamonds?
No, these are basically the fragments that map to the exact same location in the the same strand, etc. It's something we do in order to not count extra sort of non-informative fragments that come because of a technical issue.
And the other one was you said that in answer to Andrew that the commercial companies won't look at samples that have less than 5% fetal DNA.
Yeah.
What percentage have 5% or less? Is that common or is that?
Yeah, so they can detect fetal DNA from the 4th week of gestation, but most of these tests start at around the 9th week. That's sort of the optimal window, 9th to the 12th week. So from the samples that we've already collected and we've calculated the fetal fraction for, we do get, I can't exactly give you a proportion. We've only got about 100 samples collected at BCGS, but there were at least about 20 samples that had less than 8%
So
fetal fraction.
I was just thinking if it does become a clinical thing and people give their samples, how often will they say, well, you know, thanks for the sample, but we can't give you an answer. Do something else.
No, I don't have those numbers.
But it's 20%. You think would be low copy?
Yeah.
I think they're told at the moment what time period they can get tested, just like the normal, the current standard. Certain time period when you go in and get it done, but early.
I guess you get the best answer after 9 months, but then.
Yeah, we're running out of time.
So you found a sequence specificity at the cleavage site.
Yeah.
Have you used that sequence specificity to look at any of the nucleases that might be responsible for generating? Do you have any candidates?
So that's right. So we actually ran these sequences through one of these de novo motif analysis software as well. The thing was, we got these extremely short motifs, and when you got short motifs, they're not very specific, and I got a load of hits, and nothing really jumped out at me, so I didn't really pursue that avenue.
Just a general question, what's the intellectual property space like? It seems surprising to me that you could put forward another test which has already been taken up by the Children's Hospital in Great Ormond Street in competition almost with the commercial test.
Actually, because we've published what we've done, it's in the public domain, so all they need to do is just incorporate the changes we've documented. Yeah.
So you can't generate
revenue
that's going to give revenue yourself?
No.
The actual Z-score test was already published before us, so we had to make a judgment call about the novelty aspect of this. And whilst this is a great tweak, it's not the fundamental test. We weren't first to do that. What we've done is developed a nice tweak that's going to make this work so much better, right, for CMDs and lower coverage data. That's it. So we can think there was IP. Okay, last question, Matthew.
Well, I wanted to ask about those PCR duplicates as well. It may be really straightforward how those are distinguished, but doesn't that problem, when you're trying to distinguish that kind of duplicate from a real copy number variant, and you're doing it from cell-free DNA, where you say you really have to ramp up the number of cycles you do.
Right.
But does that problem become exponentially more difficult, or is it really easily
actually, no,
roughly dealt with?
The library preps have become more and more sophisticated, that if you look at the amount of PCR duplicates in cellular DNA, it's around 1%, and in the matched cell-free DNA, it's just 3%, so it's not a huge increase. So, yeah.
All right, I think we're going to wrap it up because it's past 4. Let's just thank you again for